The overall goal of this random mutagenesis is to screen for mutations that destabilize heterochromatin.
This method can help answer key questions in the heterochromatin field, such as factors that regulate the formation and maintenance of heterochromatin.
The main advantage of this technique is that it can specifically target a desired gene for mutagenesis, even if the gene is essential.
To begin, prepare yeast extract with supplements, or YES, without adenine, Pombe glutamate medium, or PMG, and PMG without adenine, by mixing the components as shown in this table. After autoclaving and adding G418 if necessary, stir the medium for another 5 to 10 minutes. Then, aliquot the media into 90 mm Petri dishes and store the plates at 4 degrees Celsius. Using the cloned RPT4 positive with its 5' and 3' UTRs and silent mutation, as well as primers P5 and P6 and a special polymerase designed to generate a high error rate, perform error-prone PCR in a total volume of 50 microliters. After generating a transformation fusion construct according to the text protocol, inoculate 10 milliliters of YES medium with yeast and incubate it for more than 16 hours to saturation. Use 200 milliliters of YES medium to dilute the cells to an OD600 of 0.2 and incubate the culture.